Have you ever stopped to think about how we get a life-saving human medicine like insulin? I mean, on a massive global scale. Well, the answer is just incredible. We've figured out how to turn some of the tiniest organisms on the planet into these powerful microscopic factories. Today, we're diving into the amazing story of how bioengineering makes it all happen. So here's the big question we're tackling. How, exactly, do you take a complex human protein, something our own bodies are programmed to make, and convince a simple little bacterium to start churning it out for us? Honestly, it sounds a lot like science fiction, but it's a very real technology that saves millions of lives every single day. And to solve this amazing puzzle, you really just need two key players. On one side, you've got the blueprint, a very specific piece of human DNA that holds the instructions. And on the other side, you've got your factory, a common everyday bacterium. Our entire job is to get that blueprint into the factory. And then, well, get production started. Okay, but first, why is this so critical? You have to remember, for millions of people around the world with diabetes, having access to insulin is literally a matter of life and death. Before this technology, the only source was from animals, like pigs and cows, and that wasn't ideal. It could cause allergic reactions and sometimes just wasn't as effective. We desperately needed a way to produce vast quantities of pure human insulin. This process is the answer.
So you might think our journey starts in a lab, with test tubes and petri dishes, but it doesn't. It actually begins on a computer. The very first step is designing the perfect digital blueprint for the job.
So scientists head to this massive public online library of genetic information, and they find the human insulin gene. But here's the thing. They don't use the whole thing. They just pull out the essential instruction part, the coding sequence. Then, and this is really clever, they basically translate it into a dialect of the genetic language that bacteria can read way more efficiently. Once that blueprint is perfect, they synthesize it into a real, physical piece of DNA. But before that gene is ready to go, we have to add one more thing. These little special sequences on each end called restriction sites. The best way to think of them is like universal connectors, like the ends of a USB cable. They are designed to plug perfectly into our bacterial machinery in the next step. All right, we have our custom-built gene ready to go. Now we need to get it into a set of instructions that the bacteria will actually understand and follow. This is where we get to do a kind of molecular cut and paste. And to do that, we use something called a plasmid. It's this small circular piece of DNA that's naturally found in bacteria. For our purposes, it's the perfect delivery truck. It's literally what we're going to use to carry our special cargo, that human insulin gene, right into the heart of the cell. So how do we get the gene into our delivery truck? It's a three-step process. First, we use these special enzymes that act like molecular scissors to cut the circular plasmid open. Now remember those universal connectors we added to our insulin gene? They create these matching sticky ends that fit right into that gap. Finally, we add another enzyme, T4 ligase, which is basically our molecular glue, sealing the insulin gene right into the plasmid. And just like that, we've created what's known as a recombinant plasmid. We've successfully edited the bacterium's instruction manual to include a brand new chapter, exactly how to build human insulin. Okay, our instruction manual is written and ready, but now we face the next big challenge. How do we actually deliver it into our tiny bacterial factories? The technique we use is called electroporation. It sounds super complicated, I know, but the idea behind it is actually pretty simple. We give the bacteria a very brief, controlled electric shock, and this little zap creates tiny, temporary holes or pores in their cell walls, just long enough for our plasmids to sneak inside. The whole process is incredibly precise. You mix your plasmids with these specially prepared bacteria, put them in a chilled little container, and then, zap! You hit the pulse button. Right after the shock, you have to rescue the cells by giving them a warm, nutrient-rich liquid to help them recover and close up those little doors. But here's the catch. This process isn't 100% perfect. Not every single bacterium in the mix will have actually accepted our plasmid. So now, we have to play detective and find the ones that did. It is time for some serious quality control. Now, our plasmid was designed with a secret weapon, a gene that makes the bacteria resistant to antibiotics. So what we do is we spread all the bacteria out on a plate that's full of an antibiotic. The result? Only the bacteria that successfully took up our plasmid have the resistance gene and can survive. The rest don't make it. It's a really powerful first filter. But
Just surviving isn't quite enough. See, it's possible some plasmids just closed back up on themselves without ever picking up our insulin gene. So we have to screen the survivors to find the colonies that not only have the plasmid, but have the plasmid with our specific insulin gene tucked safely inside. And to do this final critical check, we use a technique called colony PCR. What that basically means is we're grabbing a few cells from a colony and making millions and millions of copies of the DNA inside, specifically to see if our insulin gene is there. So, how do we know for sure we found it? It's all about this number, 350 base pairs. When we run the result of our test on a gel, we are looking for a very specific signature, a little band that shows up right at the 350 base pair mark. That size is the molecular fingerprint of our insulin gene. If you see that band, you know you've got a winner. Now we get to the payoff. We've found it, our perfect gene-edited bacterium. It is finally time to scale this whole thing up and turn this one single cell into a life-saving production line. We take that winning colony and we grow it in a huge vat of nutrient-rich liquid until we have billions, even trillions of them. Then we add a special chemical. This chemical acts like a molecular on switch, telling the plasmid it's time to start working. The bacteria start reading that instruction we gave them, and they begin mass-producing human insulin. The factories are officially open for business. Of course, all that precious insulin is still mixed up inside the bacteria, so the final step is purification. First, we use an enzyme to gently break open all the cell walls. Then comes the really clever part. We added a tiny his tag to our insulin protein, and we use special nickel beads that are designed to grab on only to that tag. And this really sums it up perfectly. The nickel beads act like a magnet for the insulin. They grab it, we wash away all the other bacterial junk, and then we add one last chemical that tells the beads to let go of the now incredibly pure protein. And there you have it. It is just an incredible journey when you think about it. We have gone all the way from a simple line of code in a database to a physical piece of DNA inserted into a custom-built plasmid, delivered inside a single bacterium, and then scaled up to produce a purified, life-saving human medicine. The whole process is absolutely amazing, and it really leaves you with one final thought. If we can teach bacteria to make human insulin, what other complex, beneficial molecules could we one day teach these tiny factories to build for us? The possibilities are, well, they're truly mind-boggling.